Hey, Biochemistry 1 students. This is a video on Unit 2 material that is designed to help you with understanding cell functions. And specifically, we're going to think about how organelles work together to accomplish a particular function. I want to also mention that I really need to contextualize this within you considering that what we are continuing to do is build upward in terms of scale. The same way that we think of how atoms interact to make a molecule, and then that molecule has some kind of thing that it can accomplish or that it does because of the way the atoms have joined together. Similar deal as we've been looking at organelles, different molecules will come together to form an organelle. Then that organelle can accomplish a particular function because of the way the molecules come together. And I also, in this video, want you to be thinking about like how multiple organelles can work together to accomplish a bigger picture function. So I'll take you through two different ones there that are directly relevant to question 2 on your Unit 2 assessment. So diving back down, that's, essentially we're going to be understanding how do organelles work together to accomplish a function, how are molecules within those organelles working together to make each organelle do what it does, and then how are atoms bound together to make those particular molecules. Okay, so it's all those interactions and the functions that emerge from them. First, what I'm going to focus on is protein production. So here is a image that shows you some of the major cell organelles that are involved in protein production. Okay. The protein production process starts in the nucleus. The nucleus contains genes, as I think you know. Genes are coded for in DNA. So DNA is the molecule whose arrangement makes up genes. Those genes are transcribed into a piece of RNA. So again, we rewrite the DNA code into a different nucleic acid molecule. And that all takes place in the nucleus through a process called transcription. When that mRNA is produced, that messenger that carries the code of a gene is produced, it can leave the nucleus, passing through the nuclear envelope by exiting through a nuclear pore. Once out of the nuclear pore, the RNA associates with a ribosome. We're going to learn these processes in more detail later, but that's the basic flow that gets genetic information from the gene, which is in DNA form in the nucleus, out of the nucleus and to a ribosome where it can be used to code for a protein. So in the nucleus, gene transcribed into RNA form, RNA leaves the nucleus through a nuclear pore, finds a ribosome, and associates with the ribosome. The ribosome reads the RNA molecule and determines what the code in that gene is instructing in terms of a protein. So the ribosome is reading the RNA and building a protein based off of the blueprint that's in the RNA. And that blueprint itself is the blueprint that came from the gene in the nucleus. So there you can see how the nucleus, which is storing the genetic information, is working together with a ribosome to make a protein. It's transcribing the genetic information from DNA form to RNA form, sending that RNA message out of the nucleus and into the cytoplasm of the cell. And then the ribosome is reading the information and using it to produce a protein. The ribosome couldn't make the protein without the DNA that is stored in the nucleus and the code that it carries. And similarly, the DNA code would be all but useless for protein production without the aid of a ribosome. All you'd have is instructions, and you'd have no machine to actually convert the instructions into the protein product. Once the protein is produced, it might just float free in the cytoplasm of the cell. But there's also the possibility that it needs to be shipped somewhere in the cell. And if that's the case, then the ribosome that produces the protein associates with the rough endoplasmic reticulum. You can see the rough endoplasmic reticulum in this model on the screen has little purple ribosomes all over it. These ribosomes are sticking to it, and they're producing proteins that go into the rough endoplasmic reticulum. The rough endoplasmic reticulum is made of phospholipids. It's a phospholipid bilayer. It's the same stuff that a cell membrane is made of that we modeled in our cell membrane lab using bubbles. The rough endoplasmic reticulum, therefore, can take the protein that has been made and it can coat it in membrane in a little bubble of membrane called a vesicle. The vesicle can then carry the protein from the rough endoplasmic reticulum to the Golgi apparatus. In this case, the ribosome is making the protein based on the genetic code that came from the nucleus. And the rough endoplasmic reticulum is then packaging the protein within membrane to ship it somewhere in the cell. So the shipping is initially done by the rough endoplasmic reticulum. The actual making of the protein is done by a ribosome. And the genetics that coded for it come from the nucleus. Once the vesicle leaves the rough endoplasmic reticulum, it typically goes to the Golgi. The Golgi contains enzymes that modify the protein so it can go off and do its function. So the vesicle fuses together in the Golgi, where enzymes then work on the protein to make it fully functional. And once the protein has passed through the Golgi apparatus, it's once again coated in membrane, shipped out the other side of the Golgi, and sent to wherever it might be going. Could be that it's going to fuse with the cell membrane, as you can see on the, on the bottom of this model. You can see a vesicle fusing with the membrane. It could be dumping protein out of the cell. Or it could go and like fuse with a lysosome, right? When I say fuse, this is like in the bubble lab where we put two bubbles together, and we saw how, something coated in membrane could pass through the membrane, or could bubble together with another piece of membrane to make a larger bubble, essentially, of membrane. If the cell in question, just to preview the next unit a little bit for you, if the cell in question, let's say, is a salivary gland cell in the glands in your mouth that make saliva. This whole process could be going on to make the protein amylase. Amylase, which I taught you about, breaks down starch, which is its molecular name is amylose. So if that were the case, let me kind of walk you through how to think about how do your salivary glands make amylase? How do they make the little protein in your saliva that breaks down the starch that you eat? Well, in the nucleus of your cells is a gene that has the right code for making the protein amylase. If that code is copied into RNA format, we call that transcribing it. It could then be exported through a nuclear pore, shipped to a ribosome. The ribosome could land on the rough endoplasmic reticulum and use the code from the nucleus to produce the enzyme amylase, which is a protein. It would then produce that protein directly into the rough endoplasmic reticulum, and the rough endoplasmic reticulum would then coat it in membrane and ship it off in a bubble called a vesicle. The vesicle would be brought to the Golgi apparatus. Again, all of this taking place in a salivary gland cell within your salivary glands in your mouth. The vesicle would fuse with the Golgi. Inside the Golgi are other enzymes that would modify the amylase until it is ready to go and do its job of encouraging hydrolysis of starch molecules within your food. When the amylase was fully modified and ready to go off and do its job, the Golgi apparatus would package it once again into a bubble of membrane called a vesicle, which would then ship to the plasma membrane, which is another way of describing the cell membrane, where the amylase would be dumped out of the cell and into your mouth. So you'd have to imagine that the outside of this cell is essentially your oral cavity. It's essentially your mouth, right? And so you'd have the salivary gland cell dumping amylase that it's making out of the cell and into your mouth, where it could then come into contact with your food and digest any starch that might be there. Multiply that by thousands and thousands of times, and you've got the way that you make amylase to break down the starch in your food within your salivary glands. And you can imagine throughout the digestive process, similar things are happening in other parts of your body, whether it's your stomach, where the cells in your stomach lining, actually they have a particular name. They're called chief cells. Um, the chief cells and parietal cells are dumping out uh, hydrochloric acid and the enzyme pepsin, which breaks down protein by similar mechanisms, just pushing it out across the cell membrane. And in the case of pepsin, the cells that secrete it are doing it using this exact same process. Nucleus has a gene that codes for pepsin, has the blueprint for how to build the enzyme pepsin, which breaks down protein in your food. That code gets copied, exported through a nuclear pore, turned by a ribosome into a protein. The protein gets packaged in the rough ER, shipped in a vesicle to the Golgi. The Golgi modifies it till it's ready to do its job of breaking down protein in your stomach. It's repackaged into a vesicle, which goes to the cell membrane and dumps it right out into your stomach. We could also keep going. The same thing is done by your pancreas to secrete enzymes that break down food in your duodenum, the beginning of your small intestine. The same thing is done by the cells in your small intestine, in your duodenum, which also make enzymes that break down your food. All of it would be this same way. If you were to look at any of those cells under a microscope, if you had a zoomed in enough look at them, you'd of course see their nucleus, but you'd also probably see that they had a lot of rough endoplasmic reticulum, a lot of Golgi apparatuses, and immense numbers of ribosomes on the rough ER. So they can crank out those digestive enzymes so you can keep breaking down your food. Of course, if you were to go to a different type of cell, like go to a brain cell in your brain, you're not going to find that it has the same amount of these different organelles because it's doing a very different job. So a brain cell, for example, has lots of mitochondria because mitochondria power the cell famously, and brain cells need a lot of energy to function. So if you go to any particular cell, you'll find more or less of these different organelles depending on the job that cell needs to do. You can think of it almost like an athlete strengthening certain muscles or certain skills in order to perform best in their particular role on a team or in a sport. Similar deal, your cells are adapted to their particular roles in your body, and they'll have more or less of certain organelles in order to do those roles to the best of their ability. Let's look at one more slightly more challenging role that organelles work together to perform within a cell. In your Unit 2 assessment, I'm calling this approximately like metabolism is essentially the role. And unsurprisingly, at the center of metabolism is the mitochondrion, right? So a mitochondrion is where carbohydrates are broken down into the carbon dioxide that you breathe out. And as those carbohydrates are broken down, the energy that's stored in them is converted to a cellular energy that cells can use. That's called ATP for all you folks with some background knowledge in cell bio out there. So mitochondria, right? They produce the energy in the cell by breaking down food. Okay, that's one way to think of them. Lysosomes are like a recycling center in the cell. Sometimes the idea is not to break something down to make food, right? Sometimes you want to break it down so you have spare parts to build something else. So it's kind of the equivalent of like if you took, let's say, an old building, you could tear down the building and burn the materials it was made of, and that would give you energy that you could use for heating or for electricity in a different place. Like, certainly you could do that. That's more what a mitochondrion is doing. It's taking your food and breaking it down, converting it to energy by essentially burning it off very carefully in a way that doesn't make fire. Otherwise, you'd catch on fire, but that does capture the energy that is in the food. Lysosomes, on the other hand, are more like taking apart an old building and recycling the parts to build something new. Okay, so lysosomes have that role. They take in old organelles that are damaged. They take in molecules that are large molecules that need to be broken down in your cell, and they do that. They break them down. Peroxisomes, on the other hand, are more of a like toxic waste disposal situation. If you have a toxin that really needs to be carefully processed, it might get targeted to the peroxisome, especially if a byproduct of processing that toxin is a molecule called hydrogen peroxide. Peroxisomes get their name from this. So they can safely process any waste that would create dangerously reactive hydrogen peroxide, and they do it safely within their membrane so that it doesn't damage your cells to process that toxic waste. On the other hand, other types of toxins are processed by the smooth endoplasmic reticulum. And the smooth endoplasmic reticulum especially does a lot of breaking down and building up lipids. So like phospholipids, for example, the molecules with the polar head and the two nonpolar tails that cell membranes are made of, those are made in the smooth endoplasmic reticulum. The smooth ER also does like alcohol detoxification. So liver cells tend to have a lot of smooth ER because they do a lot of breaking down of um, uh, any kind of toxin that enters the body, whether it be a medication, whether it be alcohol, whether it be any other toxin that enters the body through your gut. In fact, one of the interesting things about the detoxification aspect of the liver is that blood coming from your small intestine. We learned that your small intestine is where you absorb a lot of your food, right? As you absorb that food, the blood is filtered from your small intestine directly past your liver. It's the first place that it goes, essentially. And so that gives your liver a chance to filter toxins out that might have been in there. So smooth ER would be a common organelle in liver cells for this reason, because there's also some detoxification that goes on there. If you choose to answer this question, to do how do these organelles, peroxisomes, lysosomes, mitochondria, and smooth ER work together to accomplish metabolism, you'll want to read about like metabolism in a peroxisome, metabolism in a lysosome, metabolism in a mitochondrion, and metabolism in a smooth endoplasmic reticulum. And you can use keywords that I'm throwing in here, like recycling of molecules in a lysosome, energy production in a mitochondrion, detoxification in a peroxisome, detoxification in the smooth endoplasmic reticulum. If you read through those a little bit, you should find information that fits with what I'm telling you here and that allows you to give a good answer to the somewhat more challenging function of how do these organelles work together to accomplish metabolism. Okay, so for the Unit 2 assessment, the first option is to do protein production. That's the most straightforward, and I've given you a pretty solid overview of that in this video. Your second option is to take on this more challenging metabolism description. I'm giving you some hints on how you could go about that. The most challenging option would be the structural organelles. And for that one, you'd need to do some reading about the different structures that those organelles create, and then tie together how they hold cells together with each other, how they hold cells together into organ systems, like how do a bunch of cells actually form a liver is actually kind of an interesting question. It means that they're stuck together. So how are they stuck together? Well, structural organelles like desmosomes and tight junctions, gap junctions, cytoskeleton, the centrosome. These give the cells themselves structure and they hold the cells together. So if you want to take on the most challenging, I think, of the possibilities, you could try learning about those structural organelles and giving an answer based around those. All right, I hope this video is helpful to you for understanding cell functions. This is definitely a challenging topic, so feel encouraged to come in and talk to me if I can help you with question 2 on the Unit 2 assessment so that you can feel confident and comfortable about how organelles work together within a cell to accomplish a particular function. Thanks, everybody.